Hello, we're glad you've joined us for this live webinar, Querying Cytokine Signaling Networks in Myeloproliferative Neoplasms. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Fluidigm. Fluidigm develops, manufactures, and markets life science analytical and preparatory systems for markets such as mass cytometry, high-throughput genomics, and single-cell genomics. Fluidigm sells to leading academic institutions, clinical research laboratories, and pharmaceutical, biotechnology, and agricultural biotechnology companies worldwide. Their systems are based on proprietary microfluidics and multi-parameter mass cytometry technology and are designed to significantly simplify experimental workflow, increase throughput, and reduce costs while providing excellent data quality. Fluidigm products are provided for research use only, not for use in diagnostic procedures. To learn more about Fluidigm, please visit www.fluidigm.com. Let's get started. You can pose questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button down at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speakers, Jennifer Frahm, PhD, and Stephen O, MD, PhD. Dr. Frahm is a Houston-based field application scientist specializing in support and training for Fluidigm mass cytometry technology. She performed her PhD research at North Carolina State University with a focus on method development for ovarian cancer biomarker quantification using high-resolution mass spectrometry. Dr. Frahm completed a postdoctoral study at the University of North Carolina at Chapel Hill on post-translational regulation in diabetic and obese mice. Dr. O is an assistant professor of medicine in the Division of Hematology at Washington University School of Medicine. He heads a translational research group focused on the pathogenesis of myeloproliferative neoplasms, MPNs. The laboratory employs a combination of human patient samples and mouse models to study the initiation, development, and progression of MPNs. Dr. O also conducts clinical research investigating novel targeted therapies for MPN patients. He completed his MD and PhD at the Northwestern University Feinberg School of Medicine, then completed a residency, fellowship, and postdoctoral fellowship at the Stanford University School of Medicine. Dr. Frahm will now begin her presentation.
Thank you, Judy. Good morning. Thank you for attending this webinar. I'm going to provide a brief introduction to mass cytometry. As we all know, biology is heterogeneous. Cells express a complex array of proteins indicating physiological status, phenotype, and function. In order to fully characterize the heterogeneity present, we need high-dimensional technologies that enable single-cell analysis with many different parameters. The Helios is a third-generation technology. It's the only single-cell platform that can measure over 50 parameters simultaneously. These features allow one to fully characterize the heterogeneity present in biological samples. There's 5 elements of mass cytometry. The first element being metal tags. So we use metal tagged antibodies to probe the cell's phenotype and functional status. The Helios can resolve isotopes of different masses with minimal signal overlap. And as shown in the mass spectrum down below of the slide, the tags have very similar intensities. All of these features combined facilitate high-dimensional analysis and panel design. As I mentioned, we use rare earth metals to tag to antibodies and probe cellular function and phenotype. The metal tags are rare earth metals, which means that these are not found biologically, not found naturally within cells. And so there's no worry of cellular background. We have over 600 pre-conjugated antibodies in our catalog. These encompass both human and mouse specificities as well as phenotyping and functional applications. If the antibody of your interest is not in our catalog, we have conjugation and services available in which we will conjugate a metal tag to an antibody of interest. We also have metal conjugation kits available in which you can do the conjugation yourself. In addition to our antibodies in our catalog, we also have cell ID reagents. And these include a DNA intercalator, which helps us identify single cell events. We have a live dead indicator, cisplatin, an S-phase indicator, and we also have barcoding reagents. Barcoding allows one to combine 20 different samples into one tube and run them at the same time. This significantly reduces variation from sample handling and also any instrument variation that may be present. So after you decide on your panel with your metal tagged antibodies, you then stain your cells with protocols. We have protocols available, and many of these protocols are very similar to what you would do in flow cytometry. So after the cells are stained with metal tagged antibodies, a single cell suspension is then injected into the mass cytometer where the Helios ionizes each cell. The Helios uses time-of-flight technology that focuses isotope tags into discrete bands. These bands are resolved by the detector within 1 Dalton resolution, thereby isolating probe signals to individual channels with very little overlap. There's 135 channels that can be measured today, and so this encompasses all of the metal tags that we have available, as well as allows for room for more tags as they become available. The Helios generates multiparametric data for a large number of cells per sample, and this allows you to fully characterize the sample heterogeneity on a single cell basis. Since 2008, we have over 200 publications using mass cytometry. This number is steadily increasing, and many of these publications cover a wide range of research. And Dr. O will go into one of these areas related to cytokine signaling further. Thank you for your time.
Thank you, Dr. Frahm, for that informative presentation. I will now turn the presentation over to Dr. O.
All right, so first I'd like to thank LabRoots and Fluidigm for hosting this webinar and for the opportunity to present my research today. The title of my presentation is Querying Cytokine Signaling Networks in Myeloproliferative Neoplasms. And this is a general overview of my talk, so I'm going to spend a few minutes covering some background and discuss the role of aberrant signaling in myeloproliferative neoplasms, or MPNs. And then I will spend quite a bit of time talking about how we've used mass cytometry to interrogate altered signaling networks in the MPNs and how that has led to our identification of hyperactivated NFKB pathway signaling in myelofibrosis and secondary acute myeloid leukemia. And then lastly, I'm going to tell you about some very recent work where we're trying to better understand the interplay between inflammatory cytokines and how they interact with the dysregulated signaling that we have observed in the MPNs. So just some very basic background. First of all, we work on myeloproliferative neoplasms in my lab, and this is a schematic showing the organization of the different myeloid neoplasms. And there are actually several different types of MPNs, but we're primarily focused on these three MPN subtypes: polycythemia vera, or PV, essential thrombocythemia, or ET, and primary myelofibrosis, or PMF. And These three diseases share several clinical features, but they are distinct in other ways as well. And that is delineated a little bit on this next slide. So in the case of polycythemia vera, the cardinal clinical feature is increased red blood cells or an elevated hemoglobin measured clinically, and in the case of ET, in sort of analogous fashion, increased platelets. Both of these diseases, patients are at risk for thrombosis as well as paradoxically bleeding. And in both cases, for PV and ET, patients are at risk for eventual transformation to secondary myelofibrosis. Myelofibrosis can also occur de novo, in which case it would be considered primary myelofibrosis. And this disease is fairly complicated, but comes with a number of different symptoms and clinical features, including, of course, underlying bone marrow fibrosis, constitutional symptoms which can be quite severe, extramedullary hematopoiesis, and splenomegaly. It's well established in myelofibrosis as well as in the other MPNs that inflammatory cytokines, including TNF-alpha, are overproduced, and it's thought that these cytokines play a role in the pathogenesis of these diseases, though it's not completely understood exactly how they contribute. In the case of myelofibrosis, patients with this disease can evolve to secondary acute myeloid leukemia, and patients with PV and ET can go through a myelofibrotic stage and then develop secondary AML, but they can also directly transform to secondary AML. The prognosis for myelofibrosis is limited, and for secondary AML, it's somewhat dismal, in contrast to PV and ET, where overall the prognosis is much more favorable. These are a group of diseases where certainly more effective therapies are needed. So in terms of the underlying pathogenesis of these diseases, MPNs are clonal disorders that are thought to originate at the hematopoietic stem or progenitor level. So this is a schematic of the hematopoietic tree and meant to illustrate that while these diseases originate in primitive cells, they do encompass the entire myeloid lineage, such that clinically what we see, as I mentioned in the case of PV, for example, elevated hemoglobin or increased red blood cell production, or in the case of ET, increased platelets. The molecular drivers of these diseases in part has been attributed to a mutation in the JAK2 tyrosine kinase, so this is the JAK2 V617F mutation, which is known to be present in many patients with MPNs. And then more recently, other genes that are recurrently mutated in these diseases include MPL, which is the receptor for thrombopoietin, and most recently, mutations in the calreticulin gene have been identified in a subset of patients with ET and myelofibrosis. These three groups of mutations share in common an ability to lead to hyperactivated JAK-STAT signaling. So this is a schematic of the JAK-STAT signaling pathway, and I've denoted the JAK2 V617F mutation as one molecular driver of activation of this pathway. And what happens in the presence of JAK2 V617F is this pathway becomes either completely cytokine independent or at least cytokine hypersensitive, such that downstream signaling is overactive, ultimately leading to increased cellular proliferation, and in the case of PV and ET at least, increased blood counts. So this is when the JAK2 mutation was discovered in 2005, it was an obvious target for therapeutic intervention, and a number of small molecule inhibitors of JAK2 went into development. And one of these is the drug ruxolitinib, which was approved, FDA approved in 2011 for the treatment of myelofibrosis, and several years later for the treatment of patients with PV. So what we have seen clinically with ruxolitinib and similar results in general with other drugs in its class is that the drug is very effective at two things in particular. So that the top graph here is showing spleen volume reduction, and in this study, patients were randomized to treatment with either ruxolitinib, shown in orange, or placebo, shown in blue, and each vertical line represents an individual patient with their relative change in spleen volume following treatment with ruxolitinib or placebo. And what was seen in this, as well as other studies, is that almost every patient has at least some degree of spleen shrinkage, whereas with placebo, as expected, if anything, the spleen increases in size over time, which is the natural course of the disease. Once again, we're talking about myelofibrosis for this particular study. In the bottom graph, it's showing symptom response to ruxolitinib versus placebo, and there's a pretty dramatic difference here. With placebo, of course, you would not expect any improvement in symptoms, but with ruxolitinib, there was a substantial improvement in patients treated with that drug. And so this has been validated across a number of studies and in clinical practice that ruxolitinib is very effective as far as symptom improvement, and has become a very valuable treatment option for patients with myelofibrosis. So that's all well and good. However, ruxolitinib and the other drugs in this class do not really appear to be capable of eradicating the underlying malignant clone. And by that I mean if you measure the JAK2 mutant allele burden, let's say it is 80% before treatment has started, and then remeasure that, say, 6 months after starting treatment with ruxolitinib, it may have come down a little bit, say to 60% or 50%, but it will not go down to zero. And the other issues, such as the underlying bone marrow fibrosis and the anemia that often occurs in patients with myelofibrosis, these things do not go away. So we're certainly not putting the disease into remission, and really it's largely symptom improvement that is observed with treatment with ruxolitinib. So the obvious question in terms of the underlying pathogenesis is why? You know, you have a target here, you have a pathway that's clearly activated, and drugs that can target the pathway, so why are these drugs not more effective? Well, one potential reason for this is that if we go back to our JAK-STAT signaling cascade and ask ourselves, well, what else is going on? You know, perhaps we need to look at this sort of signaling network as something a little bit more complicated. So here is still a simplified schematic, but it gives you an idea of really how there are many signaling pathways which can interact with the JAK-STAT pathway, or which could be independently activated through other upstream mechanisms. For example, NFKB, one canonical activator of NFKB signaling is TNF-alpha. And again, these pathways are all interrelated in different ways. And so it's certainly conceivable that while you could inhibit JAK-STAT signaling with a targeted inhibitor of JAK2, there may be other parallel pathways which are contributing to the pathogenesis of these diseases. So our goal broadly when we started this work with mass cytometry was to really get a comprehensive understanding of these signaling networks in patient cells from these patients with myeloproliferative neoplasms so that we could better understand what's going on and then put that in the context of treatment with these targeted inhibitors. So I'm taking the same schematic here on the next slide and now outlined in white. Each of these individual proteins are targets for which there are validated antibodies that have been used for either flow cytometry and or mass cytometry. Most of these we have incorporated into our quote-unquote standard signaling panel and utilized to interrogate signaling in MPN patient samples. And Jennifer gave a nice overview of mass cytometry, so I won't go into detail, but to just give you an idea, this is the first large experiment we did with mass cytometry to characterize signaling in these diseases. And in this experiment, we had several normal controls and then 4 patients with myelofibrosis and 5 patients with secondary AML. And we had, as outlined below, about 15 surface markers that would allow us to resolve all of these different cell subsets. So when we did this kind of experiment, then we wanted to identify these populations of interest, and we used the algorithm SPADE, which I won't spend time going into the technical details, but it's one of several different algorithms that's now available to analyze the multidimensional data that's produced by the CyTOF. This just shows that with SPADE we can utilize its ability to cluster cells based on similarity of expression across the different surface markers in our experiment. And then as an example shown on the left here, I've drawn a circle around what is the stem and progenitor compartment, which is in part grouped together based on CD34 expression. There's 3 other examples on the right showing other cell populations which are pretty easily identifiable by the SPADE algorithm, and then confirmed based on expression of markers we would anticipate to be expressed in these particular compartments. So we now have these data sets with these patient samples where we can focus on these individual cell compartments and then study signaling within them. So when we did this first sort of screening experiment and looked at signaling in these patient samples, first stop was to really look at JAK-STAT signaling. And in these examples, I'm reducing it down to just looking at phospho-STAT3 on the y-axis and phospho-STAT5 on the x-axis, and first start by showing you normal. So on the plot on the left, in the basal setting, basal condition, there's very little phospho-STAT3 or phospho-STAT5 as expected. And then following stimulation with thrombopoietin or TPO, we see that there's a subset of cells that are now positive for phospho-STAT3 and STAT5. So that's our normal signaling response. And then when we compare it to the patient samples, here I'm showing 3 examples of patients with myelofibrosis and the corresponding basal and TPO-induced signals. And what I'm illustrating here is that the signaling responses are variable, and they diverge from patient to patient. And in some cases, they are hypersensitive to the TPO stimulation, meaning a higher degree or a higher level of phospho-STAT3 and phospho-STAT5, as shown on the top right. But they range down to the bottom right, where, in fact, that particular sample looks somewhat hyposensitive compared to the normal. And this, in part, reflects the heterogeneity of human disease. There is also likely to be some technical aspect in terms of sample quality, but I think it suffices to say there's considerable variability in the signaling responses that we observe. And then just to show some examples from secondary AML, essentially we see similar findings in terms of the diversity of the signaling responses to TPO. So that's to illustrate the heterogeneity, but then when we look at these, all of the samples in this particular initial experiment, and we create a heat map, now we're focusing, what I should mention, we were focusing on the prior slide as well as this slide on the CD34 positive stem and progenitor cell compartment. And in this heat map, we're showing all of the intracellular signaling molecules that were included in this experiment, and then going down vertically from the normal to the MF to the secondary AML samples. So everything is, in terms of the visualization, is normalized to the first normal donor, the top horizontal row, and again, everything is relative to that. So when we look across the sort of this broad survey of these intracellular signaling markers, what we found, first, looking at JAK-STAT signaling, while again there's significant variability or heterogeneity, there did seem to be a theme of elevated STAT5, phosphorylated STAT5, indicative of hyperactivated JAK-STAT signaling, as we would expect. But perhaps more surprisingly is that we saw a number of other signaling markers which appeared to be higher in the patients versus normal. So as outlined in orange, markers that are representative of the MAP kinase and PI3 kinase pathways, and then outlined in green, IκB alpha, which is part of the NF-κB pathway. So that was something where we weren't necessarily expecting to find anything specific, and once we did see something, obviously it warranted further investigation. Also, just to make a comment on several of these signaling markers which were elevated in the stem and progenitor cells from the patients with myelofibrosis or secondary AML, we asked whether these signaling abnormalities were specific to the stem and progenitor compartment, were they present in other cell compartments too. And here in these heat maps going from left to right, we're showing increasing levels or increasing myeloid differentiation. So this illustrates with the signals being elevated across the board in many cases that these signaling abnormalities are present across myeloid development. Here is shown as an example phospho-STAT5, and here's phospho-ERK, phospho-CREB, and then ICAPB alpha. So many of these signaling abnormalities persist from the primitive cells throughout at least early myeloid development. We also, in this experiment, looked at the response to ex vivo ruxolitinib exposure. So in these histograms here, first we'll look at phospho-STAT5 response to TPO, and we're looking at the myelofibrosis and secondary AML samples. And again, this is focused on the CD34 compartment. When we compare that to what happens in the presence of ruxolitinib, we found that ruxolitinib effectively blocked almost completely the STAT5 phosphorylation. So it was very effective, as we would expect, since ruxolitinib is a JAK2 inhibitor and STAT5 is downstream of JAK2. In contrast, when we looked at the ICAPB alpha levels, which were elevated basally in many of the patient samples, and then we asked whether ruxolitinib affected that, in most of the patient samples there was little to no effect from the ruxolitinib. In other words, the ICAPB alpha abnormally high levels were insensitive to ruxolitinib. So that really caused us to look more closely at the NFKB pathway, and here's kind of a blown up illustration to focus more on that. And on the right side is the canonical NFKB pathway, and shown in orange is ICAPB alpha. So as I'm sure many are aware, ICAPB alpha is actually a negative regulator of NFKB activity, and its role is to prevent NFKB from the NFKB subunits such as RELA from acting as transcription factors in the nucleus. And so when we think of ICAPB as a negative regulator of the pathway, when we put that into the context of observing basally elevated levels of ICAPB alpha, we were a little confused as to what that might mean. In some respects, the natural inclination would be to assume that that represents a downregulation of NFKB signaling, but we actually postulated that it was possibly the opposite, which is to mean that we know that upstream activators of this pathway, such as TNF-alpha, are elevated in patients with myelofibrosis, and this results in activated NFKB signaling, but then it also results in induction of negative feedback through the increased production of ICAPB alpha. So one could certainly imagine that in the face of chronic exposure to elevated TNF-alpha, that in the basal or sort of chronic setting in which we measured ICAPB alpha, those elevated levels of ICAPB alpha could in fact be reflective of chronic hyperstimulation of NF-κB signaling. So, we didn't really know entirely what to make of it at the time, but that was some of the ideas that we were thinking of. And we also had to consider the possibility that NF-κB could be activated or dysregulated occurring through either or both of a cell autonomous mechanism or non-cell autonomous mechanism. So cell autonomous meaning within the same cell, it could be crosstalk from JAK-STAT signaling leading to NF-κB activation, or in a non-cell autonomous fashion, you could imagine that TNF-alpha could be produced by one cell and then go and act on another cell to activate NF-κB. And it's well established that TNF-alpha is a downstream transcriptional target of JAK-STAT signaling as well, and so that seems like a very plausible scenario. So we wanted to try to sort that out, and to do so, the first step was to modify our mass cytometry panel, and we then added in an antibody for the P65 RELA subunit of NF-κB, an antibody for the phosphorylated version of this protein. And then we went back and did similar to experiments to what we had done initially, and what we found, as shown in these example plots on the left here, was that the levels of phospho-P65 were substantially elevated in the CD34 positive cells from patients versus controls. And so shown here is two patients with myelofibrosis compared to normal. And here's an example with a secondary AML patient compared to normal. So varying degrees, but some degree of increased phospho-P65 was found in many of the patient samples. And in response to TNF-alpha, we also found that many of the patients had a hyper response to TNF-alpha in terms of even higher levels of phospho-P65 stimulated by TNF-alpha than what was seen in the normal. So we did this across a series of approximately 20 patients with myelofibrosis and secondary AML, and this is summarized here, and compared against all of the normal controls that we used in these experiments. And we were able to show that basal phospho-P65 levels were significantly elevated in both myelofibrosis and secondary AML. And there was also evidence, though not statistically significant in myelofibrosis, that in the presence of TNF-alpha, we also found elevated phospho-P65 levels compared to normal. So this data, provided pretty clear evidence that there was NF-κB hyperactivation in CD34 cells from these patients with myelofibrosis and secondary AML. I'm not showing it here, but we corroborated this with gene expression analysis using GSEA in a published data set, and found also a upregulation of NF-κB downstream gene targets in CD34 cells from patients with myelofibrosis. And then we wanted to understand the relationship between NF-κB hyperactivation and JAK-STAT signaling. In other words, is NF-κB being activated as a downstream consequence of JAK-STAT hyperactivation mediated by JAK2V617F or some other driver mutation? So we did a simple correlation analysis between levels of phospho-P65 and phospho-STAT5, and shown here there was some degree of correlation in both myelofibrosis and secondary AML. And then we also utilized the conditional probability algorithm DREMI, which looks on a single cell basis at the relationship between phospho-STAT5 and phospho-P65. And what we saw, as summarized in the graphs on the right, is that there was a significantly higher DREMI score reflecting a higher essentially linkage between phospho-STAT5 and phospho-P65 in the patient samples compared to normal, indicating that there at least is some relationship between the two, though it does not specify if it's through a direct interaction or through perhaps secondary mediators. We also asked the question, as I kind of alluded to earlier with IκB alpha, as to what are the effects of ruxolitinib. And so this is ex vivo exposure to ruxolitinib, and here we're looking at the effect on phospho-P65 levels, again focusing on the CD34 cells. And in a nutshell, what we found in both MF and secondary ML was that the ruxolitinib did not substantially reduce the phospho-P65 levels. So consistent with the earlier experiments with IkB alpha, it appears that this abnormal hyperactivation of the NF-κB pathway is not ameliorated by JAK2 inhibition. Those experiments were with relatively short-term exposure to ruxolitinib, about 1 hour, and we wanted to see if longer exposure to ruxolitinib would perhaps have a greater effect. And so we did this through a variety of different time course experiments up to 6 or 12 hours. The normal controls are shown at the top here. It's kind of difficult to see, but we basically found that in comparison to the normal donors, in the case of the patients, ruxolitinib did not fully abrogate the elevated levels of phospho-P65. And interestingly, just looking at the basal condition and comparing within a patient longer times in culture, so in the case of the sample shown on the left, we are looking at 1 hour versus 3 hours versus 6, and on the right, 1 versus 12 hours. What we found is that even just in the basal setting, meaning these cells are just in culture in the incubator for that period of time, the phospho-P65 levels went up substantially. And that would be potentially consistent with the notion that these cells are producing TNF-alpha, hyperproducing TNF-alpha, and the longer you leave them in culture, the more that effect is seen in terms of just sort of basal or baseline phospho-P65 levels. So coming back to this schematic as far as cell autonomous versus cell non-autonomous, so again, we wanted to better understand that, and so one way to look at that is to survey the cell populations that are affected in terms of these abnormalities in the NF-κB pathway. So here are some spade trees looking at the levels of phospho-P65, and this is a little bit hard to make out, but on the left side we're looking at a normal control, and on the right a patient with myelofibrosis. And these spade trees are color-coded for phospho-P65 levels, and then the stem progenitor compartment is outlined here. But the main point here is that in contrast to the normal cells, in the case of the NF sample, we see at least some degree of NF-κB or phospho-P65 levels being elevated pretty broadly across the entire hematopoietic tree. And this includes T cells, which are not myeloid cells. And for the most part, the T cells are thought to not be involved in the malignant clone, meaning they do not harbor the mutations such as JAK2V617F. So therefore, specifically in the T cells at least, if there is an abnormal activation of NF-κB, it would imply that it's occurring through a non-cell autonomous mechanism, since those cells don't actually even harbor the malignant mutations. Here is another example with an AML case showing a similar broad distribution of NF-κB signaling, more so than in the normal control. And then here is looking at another example specifically focusing on the T cells, as shown on the bottom right in the particular patient with MF, indicating the difference between the patient and normal. And then we look also at what happens in the context of stimulation with TNF-alpha. Here, as shown by the red arrows, the most robust NF-κB activation is occurring in the CD34 compartment, both in the normal and in the patient. So that illustrates the point that that context matters in terms of the condition. So basal versus TNF stimulated or other perturbations may give different results, which inform the interpretation of what's actually happening or what could be happening in vivo. So coming back to TNF-alpha, I mentioned that this is one of several inflammatory cytokines which are known to be elevated in MPN patients, and most particularly in myelofibrosis. And we did our own analysis looking at circulating plasma levels to confirm that, utilizing many of the same samples that we examined by mass cytometry. And this graph just shows that we did find that TNF-alpha levels were significantly elevated in both myelofibrosis and secondary AML. But we wanted to understand where the TNF-alpha was coming from, because that's plasma, so total plasma levels of TNF-alpha. And so we took advantage of the ability of mass cytometry to measure intracellular cytokine levels, and we developed a separate antibody panel where we measured about 15 intracellular cytokines in conjunction with the surface markers to identify the different cell subsets. And then we could identify where these cytokines are being hyperproduced. And so these spade trees are pretty difficult to make out, but the point that is illustrated here is that in the case of TNF-alpha, which is being shown here, as well as many of the other cytokines, the highest production of the cytokines was occurring in the monocytes, and to a lesser extent in myeloid progenitors and the stem progenitor compartment. And we looked at that more closely. So in these examples on the left, the plots are showing monocytes, on the right lineage negative CD34 cells, and looking at one normal versus two patients with myelofibrosis. In the basal condition shown in the top rows, there were only a small percentage of cells which were positive for TNF-alpha, although that number was at least slightly to significantly higher in the patients compared to the normal. We also looked at the levels of TNF and the other cytokines in the presence of PMA ionomycin, and we found that although the normal cells responded as expected with an upregulation of TNF-alpha production, there was a hyper-response, higher levels of TNF-alpha in the context of PMA ionomycin in patient samples as well. That is summarized on the bottom left graph, indicating, again, both in the monocytes and CD34 cells, we saw increased levels of TNF-alpha. We also looked at this in a cell line, the BAF3 cell line expressing MPL as well as JAK2 V617F versus wild-type JAK2 versus parental cells, and found that expression of JAK2 V617F was sufficient to lead to substantially increased production of TNF-alpha, providing further support for that specific mechanism to be in play, that JAK2 V617F leads to increased production of TNF-alpha, which then activates NF-κB. So, putting the signaling and the cytokine data together, we asked ourselves, well, are these things happening in the same or different or overlapping cell compartments? And on the left, we're looking at phospho-P65 levels in various cell compartments, and we found that, as I showed earlier with the spade trees, in the basal setting, we see a broad distribution of NF-κB signaling. In the presence of TNF-alpha, it's a little bit more enriched in the more primitive cells. And in contrast, in terms of TNF-alpha shown on the right, really most prominently overproduced at the highest levels in monocytes, so indicating that it may be that the monocytes are what produce the bulk of the TNF-alpha, and then they go and act on more so the primitive immature cells. We looked at the effect of ruxolitinib on TNF-alpha levels, and on the left, this is looking at plasma levels of TNF-alpha and breaking it down in terms of patients with myelofibrosis pre or post-treatment with ruxolitinib and comparing that to normal donors, and we found that the levels pre-ruxolitinib were elevated as expected, and in the presence of ruxolitinib treatment, this is therapeutic administration, so in vivo, the levels remain, they may have come down slightly, but they remain significantly elevated compared to normal. And this is consistent with published literature, just further establishing that TNF-alpha levels are not normalized by ruxolitinib. So we wanted to look at this in the individual cell populations that we examined by CyTOF. And we, here I'm showing monocytes, and again, it's a little bit hard to see these graphs, but it's quite clear that in the presence of ruxolitinib, the elevated levels of TNF-alpha in the monocytes from patients with MF, here shown two examples, were not substantially reduced by ruxolitinib. And another example in CD34 cells, the same was true, that the subset of cells that were positive for TNF-alpha did not go back to normal in the presence of ruxolitinib. So, I'm really focusing on TNF-alpha and NF-κB, but with the CyTOF, we have the luxury of being able to look at many different cytokines at the same time, and we certainly think that's relevant, given that in these patients we know it's already been established that many different cytokines are aberrantly produced. So we wanted to ask whether we could show that some of these same cytokines are being produced by, overproduced by specific cell compartments such as monocytes. So focusing right now on monocytes, and these graphs are illustrating that, again, TNF-alpha is not the only one, that many of these other cytokines are overproduced in the patient samples. So highlighting, for instance, in this patient, pretty prominent upregulation of IL-6 positive cells as well as MIP-1 beta, and in this other patient, more so TNF-alpha and interferon gamma. These are radar plots just to try to summarize or encapsulate the data we have with the cytokines, and each spoke of the wheel here is a different cytokine, and then the lines basically show the relative the relative levels of positive cells for each particular cytokine. And the left plot, each individual patient with MF is colored with a different color, and then compared to the normal or the average of the normals, which is shown in black, and the MF average is shown in red. So you can see that there is a substantial difference between the two groups, and the same data is shown a little bit differently on the right, with each individual patient colored with the same color, light blue, to try to make it visually more clear the difference between the average MF and the average normal, shown in blue versus red. So we've also done some preliminary work looking at response to different perturbations such as TLR ligands like R848 or PAM3 CSK4. And basically we see similar themes, which is that in the patients there is hyperproduction of a variety of cytokines. In this example, we're showing TNF-alpha in the two patients compared to normal. And when we look in T cells, so again, I mentioned earlier that these are not, myeloid cells are not in most patients thought to harbor the actual mutations that are present in the malignant clone. And yet, despite that, here are several clear examples, particularly in the presence of PMA and ionomycin, where there are substantially higher numbers of positive cells in the patients versus the normals. On the left, the example is MIP1 beta, on the right, interferon gamma. Okay, so just want to talk for a minute about what we're doing currently with this work. One area of interest is to understand how NFKB activation and of course other signaling abnormalities are characterized as across the spectrum of MPN disease states or disease progression. So we've looked primarily at myelofibrosis and secondary AML, but we're very interested as well as in PV, which can ultimately transform to MF and then to secondary AML. So we have some initial work that we've done with specific patients where we have matched serial samples, which has been quite interesting. And then we'll plan to look at a series of additional patients with all these different disease states. We are in sort of the follow-on work from our identification by mass cytometry of NFKB hyperactivation. We're now trying to interrogate this in mouse models and using genetic models as well as other systems to really understand what this means, the fact that we've identified NFKB activation, and is it something that we could perhaps intervene in therapeutically. And that's the next point, is really this concept of leveraging this dysregulation of this pathway. In other words, can we, rather than maybe simplistically thinking just eliminate all of the abnormality, can we actually sort of repurpose the signaling pathways to point to a selective effect on the malignant clone? So I'm going to close with acknowledgments, and several people to really acknowledge who have been critical for this work. Within my own lab, this project was initiated by a former postdoc, Betsy Engel, and has been carried on by another postdoc, Daniel Fisher. And all of the mass cytometry work we're doing currently and recently has been conducted through the Immunomonitoring Laboratory, which is part of the Bursky Center for Human Immunology and Immunotherapy Programs, or CHIPS. And I wear two hats, so I also oversee the half of the IML that houses our two CyTOF instruments. And Olga and Kate in particular have been extremely helpful for this work, as well as two students in our Masters in Biostatistics program, Hengray and Liang before him, who have contributed to the analysis of our data. And I would also like to thank my funding sources, and I will be happy to take questions. Thanks for your attention.
Thank you, Dr. Frahm and Dr. O, for your presentations. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Frahm and Dr. O will answer as many questions as time permits. And the first question is for Dr. O. What is the age range of patients with MPNs, and are there any differences in either signaling pathway activation or response to treatment associated with age?
So MPNs are generally diagnosed in patients of older age, typically ages 60s, 70s, and whatnot. But we certainly see patients who are diagnosed at younger ages as well. We have not looked exhaustively at the age of different patients and their signaling phenotypes. However, it's well known that in some MPNs, in particular in polycythemia vera, which has not really been the focus of the work that was shown today, but in that disease, there are some differences in the clinical features of patients who are either younger or older. And what's a separate project in the lab is to try to understand whether those patients have different characteristics phenotypically and genomically, and so that's something that's ongoing.
Dr. Rowe, this next one's for you too. About how long did it take you to validate your panel?
So that's a good question. I guess the short answer is for much of the work that was shown here, it was on the order of 6 to 12 months to get it really optimally validated. The longer answer would, one point is it's never done. So we're always kind of tweaking the panel, considering adding new antibodies and therefore, potentially swapping antibodies out because our panel is generally pretty full. And the work actually that's included in this presentation actually went on dates back to actually about more than 5 years ago, partly because at that time when Cytokine was kind of just taking off, there weren't really that many commercially available antibodies at the time. So there's much more customization that was required. These days, the landscape has changed considerably. There are many more antibodies that are available pre-conjugated from Fluidigm, and maybe that's something I can throw it over to Jennifer to comment on.
Yeah, thanks, Steve. Yeah, we currently in our catalog, we actually have over 600 primary antibodies. We also have 17 secondary antibodies, so if you already have something that's labeled for flow, you can maybe use one of our secondary antibodies. Most of our antibodies, I will say, are human and mouse specificities. We do have some that are cross-reactive, and I will say we do have several researchers now that are using some of the human clones that also work for non-human primate research. We also have pre-designed panel kits, and these are kits that are designed to be modular. So you can take, say, for example, a phenotyping panel kit, which is meant to ID major leukocytes. Then you can couple that with maybe a functional panel like cytokine or signaling or cell cycle kit. And then there's still many other channels that you can add in your other favorite proteins that you might be interested in looking at. So it really helps people get up and going a lot faster than the early days of mass cytometry.
Okay, next one is for Dr. O. Since the defect is present in stem cells, is there any impact on the lymphoid compartment with current treatments? And also, did you monitor that in your mass cytometry panel?
That's a good question. So, I think as particularly in the latter half of the talk, I alluded to, these are myeloproliferative diseases, so they are thought to start in the stem or progenitor compartment and then encompass the entire myeloid arm. But what we observed is that, in particular, we saw signaling abnormalities, specifically NFKB, present in T cells in the lymphoid compartment. So we think that the diseases are more complex than just looking at myeloid cells, and that there are lymphoid cells involved as well, and whether that is because in some of those patients or some of those cells there actually are the same mutations present, or actually it's more through a cell non-autonomous mechanisms. We can't say for sure based on our results, but I guess the general point is I think the important one, which is that the lymphoid cells are likely involved, or at least abnormally functioning, as a result of what's driving these diseases.
Did the cell activation or staining protocols for phosphosignaling experiments need to be modified when doing mass cytometry, Dr. O?
Yeah, that's another good question. So I didn't go into detail, but our protocols are a little bit different when we look at signaling or cytokines. And in a nutshell, when we do the signaling experiments, we use methanol to permeabilize the cells. So we do the signaling perturbations, then we fix the cells, we stain the surface markers, and then we methanol permeabilize and stain with the intracellular signaling antibodies. With the cytokine experiments, we use a saponin-based permeabilization, and we also do things a little bit differently by staining the surface markers on live cells prior to fixation. So a little bit different. Ideally, we would combine the two approaches so that we could measure signaling and cytokines simultaneously, but that is something that, at least historically, has been a little bit challenging, and because we have more than enough antibodies to fill out our panel, whether it's cytokines or signaling, we have essentially kept them separate so far.
Okay. Inaki attendee, sorry about that, thanks you for a great talk, and is curious for analysis of which program was compatible, i.e. SPADE or Citrus, Dr. O.
So yeah, so the most of the data I showed in this presentation was using SPADE, but we have used most of the fairly commonly utilized algorithms to look at our data, including VISNI, which has a lot of strengths as far as analyzing this kind of multi-dimensional data, but I didn't have anything included in this particular presentation. We've also used Citrus, mostly with a slightly different project, but we've found that to be useful in other ways as well, and we've also used Phenograph and a few other methods. So in general, I think with CyTOF, depending on the question you're asking and the types of samples available, there are a variety of different tools that could be particularly applicable.
Dr. O, how do secretion inhibitors affect this kind of cytokine data, and should they always be used?
Yeah, so it's another good question. So I think that generally speaking, cytokine or secretion inhibitors are used as pretty much a standard in these kinds of experiments. In the data that I presented, in that data, we use secretion inhibitors routinely. We've done it both plus and minus secretion inhibitors and generally gotten similar results, but most consistent data has been with secretion inhibitors, and we've stuck with that, again, primarily because that seems to be the standard approach in this field.
Did you validate your mass cytometry panels against standard flow cytometry, Dr. O?
Yes, so that's a good question. So many of the antibodies we have examined by flow cytometry as well. We didn't necessarily, in a dedicated fashion, do a side-by-side comparison as we were developing our panels, partly because I think pretty much every antibody in our panels is an antibody that has previously been validated for flow cytometry and has been sold as a flow antibody, and that's something that when I kind of wear a couple different hats, and I run the core lab that houses our CyTOF instruments, so I'm often meeting with prospective users, and when we start talking about antibodies, the first piece of advice I will give is ideally use antibodies that are already known to work for flow cytometry. So if that's the case, then then it's likely to be fine for CyTOF. But in the case where someone were to want to use an antibody where it's either not known or not validated for flow cytometry, then I think you have to ask that question as to whether it's even going to work. But generally speaking, if it works for flow, it works for CyTOF. And once again, we did validate many of the antibodies for flow, but not necessarily every single one.
Thank you. As more audience questions are coming in, I would like to once again thank Dr. Frahm and Dr. O for their presentations. Do you have any final comments for us?
Yeah, I don't know if the presentation got cut off at the end there for everybody, but the sort of take-home points with the work that I presented today are that sort of from a very broad perspective, I think I've hopefully demonstrated that mass cytometry is a very useful tool to profile, in particular in our case, hematopoietic cells where there are multiple different cell compartments. And so we can utilize the high parameter aspect of mass cytometry to include markers that can identify these different cell compartments, but at the same time have room in our panel to include either signaling or cytokine antibodies so that we can phenotypically profile these cells in terms of aspects of, again, signaling or cytokine production, which reflect their underlying cell behavior. So in a broad sense, again, this technology has been very useful for us, and in fact, in thinking about alternative methodologies, when you're looking at a single cell on a single cell level, there's not really a good alternative to be doing this with primary blood or bone marrow cells. And the more specific sort of take-home points on the scientific end with regard to the myeloproliferative diseases, which is the focus of our work, is that with this approach, we've really, I think, uncovered that signaling networks in these diseases are very broadly dysregulated and really not as straightforward as simply JAK-STAT hyperactivation. And further, with the cytokine work that we've been doing more recently, understanding that the mechanisms by which these signaling networks become dysregulated is likely to be complex, of course, but also likely to be a mixture of both cell-autonomous and cell-non-autonomous mechanisms, primarily mediated by these altered cytokines that are being produced.
Thank you again, Dr. O. Dr. Frahm?
I think Dr. O summed it up very nicely. I don't think I can add anything other than that, what he said.
Thank you again, and I'd like to thank the audience for joining us today and for their very interesting questions. Questions we did not have time for today will be addressed by the speakers via the contact information you provided at the time of registration. We would also like to thank our sponsor, Fluidigm, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through November 9th, 2017. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.